Great. Hi, good afternoon, and thanks very much for the opportunity to speak in the conference. My name's Fletcher. I'm an infectious diseases clinical academic at the Liverpool School of Tropical Medicine. I normally work in emerging infections and viral hemorrhagic fevers, but like everybody else has been distracted by COVID. And what I'm going to talk to you a little bit now for sort of 7 or 8 minutes is around the AGILE clinical trial platform. So AGILE, and as per the title here, and this wasn't a title that we developed ourselves, it was actually one given us by pharma, really, to say what you're really trying to do is act as a proof of confidence engine for novel COVID-19 therapeutics. And I'll explain why we do that in the next few minutes, a bit about the trial design and the trials that we have up and running, which are about 5. So the first question seems a relatively obvious one, especially with the news yesterday from MHRA, one of the candidates that we've had through AGILE, of why antivirals. There was clearly a big focus about vaccine rollout, as we've heard about, but we all know that the vaccine isn't perfect. And despite having highly effective vaccines, we were always going to need effective antivirals. And really, to be a game changer for health systems, we needed antivirals to use in early stage disease, to effectively stop people turning up to hospital by treating them early with oral antiviral therapy. And obviously, one of the first bills to be licensed yesterday was molnupiravir, which is a drug that we identified actually as the first candidate to come into AGILE, of which we did the phase 1 dose escalation and are now about to complete our phase 2 with intensive viral sampling. I can talk about that later as we go through. So when we think about new treatments for COVID-19, I think when we've all been in this fight for the last 18 months, we sometimes forget where we were at the beginning, and this is really where AGILE was born. And I think at the beginning, we knew when our health systems were overwhelmed that actually the main intervention was non-pharmaceutical, and it was lockdowns to limit transmission and offload the health system. So on the back of that, a collection of academics and institutions came together to form the AGILE Coronavirus Drug Testing Initiative, really. And it acknowledged this background of problem, that actually the conventional drug development pathway is not really designed for a pandemic. It has clear steps to go through, it's relatively slow, and it's not particularly responsive. We were clear that we actually needed new antiviral compounds to be called game changers, as the latest one is now called. And whilst many compounds in that first wave were repurposed, if you think about hydroxychloroquine, remdesivir, and Kaletra, to name a few as the first drugs to go into Wuhan trials, we felt that was going to be a stopgap and needed to really engage with pharmaceuticals and small biotechs to bring forward the most promising compounds. I think we must also remember, whenever we're doing these trials, that actually, particularly at the beginning, it was considered a pretty challenging and high threat environment to work in, to do early phase trials and other trials on patients with COVID at the beginning. And that includes facilities, the staff, the environment, the infection prevention and control precautions that you have to take, and recognizing, and this is an important thing, that each study, each trial, each drug carries a degree of opportunity cost. So this slide just shows through what we think is the normal pathway. And if we just remind ourselves to go from the preclinical space and a huge number of candidates being developed by pharma and take it through from phase 1 into phase 2 studies and finally phase 3, that might be in a fair wind 6 to 7 years if everything lines up. Clearly not appropriate for a pandemic situation. What we tried to do with AGILE was then to accelerate that pathway and get it down to the 12 to 24 month timeline. So what we wanted to do was sit in this early phase trial space, fast track promising candidates. And a key part of that was the ability to select our own candidates that we felt were the most promising ones, also ones that had agreements about global access, was to screen out compounds which we thought had limited efficacy quickly, to limit their use in larger phase 3 trials, to try and accelerate decision-making between phases, and the ability to seamlessly transition within the trial between phase 1 into phase 2. And then the other aim was the ability to test multiple candidates at once, which has actually proved probably the most challenging. So this is really the schematic of where the AGILE clinical trial program sits. So within the UK, it is a designated early phase clinical trial platform for COVID, in line with our sort of government support and funding. And we sit here in this space. So first in human trials, into 1B, first in patient, and then all the way up to the sort of early 2A phase, where we're gathering efficacy on that predominantly antiviral candidate. And obviously sitting quite a long way in front, but with really good communication with the trial platform's recovery in principle to try and identify promising compounds. And the way that we referred compounds up until recently was through the national prioritization platform that would designate the next compound that was referred to AGILE for consideration. And what the trial is a multi-arm, multi-stage platform trial with a Bayesian design. It has the ability to evaluate new drugs both in the community or in hospital settings, And whilst our predominant expertise and focus was on antivirals, also the ability to look at host immunomodulators too. What we wanted it to be these key things was pragmatic. So the ability to go first in human into COVID patients. Now the normal pathway doesn't allow that apart from for some exceptional drugs. So normally you would go first in human into a healthy volunteer population. We felt we didn't have time for that and had the expertise to take the right candidates with the right safety background directly into patients first up. So you start to gather safety data and efficacy in patients from the first time you use the drug. We also wanted it to be pragmatic. So with the basis of having a master protocol, we had a variety of different endpoints that could then provide a list that we could then tailor towards the individual trials. And that included also the population that we would select too. This key feature about being seamless so that we would transition between phase 1 rapidly into phase 2 pretty quickly, and then efficient. And that's really where the Bayesian approach came in. Now, Bayesian sort of statistics and Bayesian clinical trials are probably still considered witchcraft by some, but I think increasingly by regulators in early phase and later phase trials are recognized as probably the approach of the future. They don't replace the frequentist approach, but certainly they now have their recognized benefits. And so this is in the bottom right there, you can see the idea that we have a master protocol, and then from that we have individual candidate specific trials that come from that running different therapeutics. The idea at the beginning actually was to be able to run multiple different trials in parallel with the ability to share controls if the trials are similar enough. That proved pretty challenging, and most of the trials have not been able to do that due to different inclusion criteria and slightly different mechanisms of actions of the drugs really. So this is really how the trial actually works in practice. So we identify patients that test positive for COVID-19 in the UK or overseas. They're then brought into screening, so out of their isolation period, consented, and then randomized. And I think we can't underestimate the fact that what you're doing is bringing a patient, particularly at the beginning, who was relatively unwell with COVID, and then consenting them potentially to have a first-in-human drug with extremely limited safety data in a human population. So that's not a small undertaking. Patients are then randomized between placebo, which is normally the control arm, occasionally standard of care, and then the actual IMP. And the way that the trial works is sort of demonstrated by this circle. So we look at small numbers of patients within phase 1, where we're really trying to confirm the dose and the safety around that new compound. And that might be a small number such as 6 patients at the first dose, of which 4 would receive drug, 2 would be on placebo. And we cycle them through until they get to about day 7 of receiving the treatment. And we then stop and review safety at that stage with the ability to go to a safety review committee and then dose escalate normally about 2 weeks later. At the same time, the patients continue in the trial to at least day 29 so that we get more in-depth safety data. And that sort of timeline can be flexed depending on the drug that's being used. And what we might well do for a different trial then is dose escalate 2 or 3 times up to what we think is the optimum dose in this sort of cycle of 6 patients, often with a ratio of 4 to 2. What that enables us to do is then do dose selection and then move seamlessly after we've confirmed dose into a phase 2A trial where we're trying to define the efficacy of the drug. And what we then do is have interim assessments where we look at efficacy of the drug and either drop that candidate through futility or progress it on to the end of the trial, or stop the trial for efficacy and then refer it on to one of the later phase clinical platforms for evaluation. This is just one of the schematics that we have for a trial that we run overseas. So as well as recognizing that AGILE should operate predominantly in the UK, supported very well by the UK CRF network, which many might not know, but is a network of trial centres supported often by NHR that provides access to expertise in early phase clinical trials, and they've been a big support to AGILE. However, look, with our global outlook, we were quite keen on the ability to be able to run trials overseas. Now, running early phase trials in some settings is pretty challenging, but working with partners, particularly in South Africa and Uganda, we have this capability. What we didn't want to do necessarily was to do a first in human drug overseas to a degree of reputational risk and the optics of that. And so actually within the platform, we have the ability to do first in human trials in healthy volunteers too, and then rapidly escalate them into a 1B in patients overseas. Now, I'm just going to cover really quickly actually how the Bayesian model works. So we think in summary, it's quite an efficient way of using information by using a prior distribution, then using the data to get a posterior. And what we think it does, and the trial design is supported by Thomas Jackie and others in Southampton who've evaluated this, is it accelerates our ability to make decisions. What we really aim to do within phase 1 is establish the safe dose And we do that by means of dose-limiting toxicities, and that's what we call, a grade 3 level adverse event, or SAE. And what we do is set the level at this level here. So it's a 25% chance that treatment gives us an increased risk of dose-limiting toxicity over control, and then run that through the model. And this is actually what it looked like when we ran molnupiravir through. We were the first group. We were following slightly behind a healthy volunteer study being run in the UK, but we were just behind that, running this through up to the optimum dose of 800. And as you put data in, the model is changed as it accumulates safety data between the different cohorts. And this is essentially what it looked like for molnupiravir as we ran this through the platform. However, what we also want to do is use a Bayesian approach to demonstrate early efficacy. So we never thought that AGILE would function as a trial that demonstrated conclusively that drug A or drug B was better than a control or others. But what we wanted to do was give that early signal of efficacy to really give it the rationale and the justification for evaluation in later phase trials. So what that means is often we don't have the sample size, particularly now in a vaccinated population, to demonstrate any clinical endpoints. So it's mainly virological related to antivirals. And that's built in interim analysis around futility and efficacy, as is shown, which then allow us to stop the trial early for either of that reason, or potentially carry on to the end of the sample size. And then lastly, this slide, there was another slide that seems to have slipped out where I talk about the individual trials that we're running, but we've obviously received funding from government, MRC, Wellcome Trust, and then from individual pharmaceutical companies, with the line that actually, if you're bringing your pharmaceutical drug to us, we sort of expect you to pay for that drug development through AGILE whenever possible. And the trials that we have up and running now are 5. So our first trial that we prioritized was molnupiravir, which is quite a long time ago now, when we did the phase 1 dose escalation trial of that, and then are about to finish a phase 2 with our independent DMC on Monday, actually, where we hopefully will have adequate data to stop that trial as well. And that has pretty intensive virological surveillance. The second trial in is high-dose nitazoxanide, of which, again, we've done the phase 1 and the phase 2 is running in South Africa. The next trial, the third trial that's up and running, is a monoclonal between GSK and Vir, and we're at the end of the third cohort of dose escalation of that pretty promising monoclonal, we hope, before we transition into a phase 2 in a couple of weeks. And then lastly, we have intravenous favipiravir, which has been prioritized by UK CTAP, which will go into a hospitalized cohort in the next couple of months, and inhaled niclosamide as an outpatient group. And that's currently all the drugs that have been selected to run through AGILE that are at different stages of development. Okay. And then lastly, I think I'm just about finished with time. Obviously, we have a number of sites that operate through the UK. Southampton is the coordinating trials unit for our trial, and then we run it in Liverpool, Manchester, Lancaster, and London now have come on board as well, with multiple different people involved, as well as our overseas sites, predominantly in South Africa, but also in Uganda.